Dear students, now we are going to discuss a dark phase, dark reaction, that is a second part of photosynthesis. So this is a second part. So here, dark reaction is not a misnomer. It is not a misnomer. Misnomer means that gives us a wrong meaning. Dark reaction. Dark reaction means that phase takes place even in the absence of light. Here, according to this, dark reaction takes place dark only? No. The word dark refers to it is independent of light, depends on end products of a light reaction. So indirectly it depends on light phase. Directly it is not dependent on light phase. So to reflect that meaning, they use a dark phase. So we should not construe that the dark reaction is taking place in dark conditions only. That is wrong one. Misnomer means a word that will give us a wrong meaning. Even though the word was based on the base of a particular meaning, it will give us a wrong meaning. So it looks like a misnomer, but it is not a misnomer. Here, dark reaction means it is indirectly dependent on light. Directly, it is independent. Possible in the absence of light if ATP and NADPH plus available. For example, light available, dark reaction is initiated. Light is not available, dark reaction is stopped. So it depends on end products of light. So during non-cyclic electron transport system, where H2O is acting as hydrogen donor and electron donor, what are the end products of that oxygenic photosynthesis? ATP, NADP, HH plus, and oxygen. So oxygen diffuses out of the chloroplast and available within the plant body and available in the environment. And what about the fate of ATP and NADPH? So where are the ATP and NADPH H plus utilized? That is during dark reaction, right? So in dark reaction, on the base of a first stable compound, number of carbon atoms present in the first stable compound. So dark reaction divided into C3 cycle. C3 cycle. And the second one, C4 cycle. Majorly two kinds. On the base of a number of carbon atoms present in first stable compound after carboxylation. If it is a 3-phosphoglyceric acid, a 3-carbon phosphoglyceric acid, that is acid, not a sugar, the consent cycle is a C3 cycle. If it is a 4-carbon organic acid, OAA, oxaloacetic acid, it is a C4 cycle. So what was the criteria on the base of which they classified a dark reaction into two categories on the base of number of carbon atoms present in first stable compound after carboxylation, right? So we'll see C4 cycle later. Now what about a C3 cycle? So C3 cycle was the carbon path during C3 cycle was traced out by Melvin, Kelvin, Basham, and Bensham with the help of radioactive isotope, that is a 14CO2, we studied at the time of introduction part of physiology. 14CO2 in the case of chlorella, an algal member. With the help of algal photosynthesis, by giving 14CO2 and other raw materials, they prepared a flowchart of carbon reduction in a plant, that is chlorella, that is common for all photoautotrophic plants. Okay, so now what about synapse, main areas for C3 cycle? So in C3 cycle, first stable compound is phosphoglyceric acid, PGA. Here I'm writing PGA. What is PGA? Phosphoglyceric acid. It is a 3-carbon compound. So after carboxylation reaction, we will see what is a carboxylation reaction. The first stable compound is a 3-carbon compound. Hence the cycle C3 cycle. And place of a C3 cycle is occurrence. It take place in. And before that one, it is a biosynthetic pathway. Biosynthetic pathway. So what is that a biosynthetic pathway? During C3 cycle, CO2 is reduced to carbohydrate sugars. Here I'm writing carbohydrates. Okay, so C3 cycle is only biosynthetic pathway. This is the only biosynthetic pathway present in all photoautotropes. So this statement is very, very important. So C3 cycle is the only biosynthetic pathway present in all photoautotropes or autotropes, particularly photoautotropes. Autotropes also correct. Autotrophs. Even for bacteria, all those which are able to reduce a C of CO2 into carbohydrates, this is the only biosynthetic pathway. So this is common for all photoautotropes or autotropes which are able to convert a C of CO2 into sugars. Carbon assimilation pathway. Carbon is assimilated. What is assimilation? Conversion of a inorganic form of carbon. This is CO2, inorganic form of CO2 into organic form, complex organic form into sugars. So this pathway is common for photoautotropes. This is the only mechanism by means of which so-called photoautotropes are able to reduce the C of CO2 into carbohydrate. All photoautotropes, cyanobacteria, blue-green algal members, bacteria, chlorobium, chromatium, rhodospirillum. So all these, all eukaryotes and prokaryotes, those which are able to perform carbon reduction, C of CO2 into sugars, that is whole and soul by means of a C3 cycle only. Next, place of occurrence of a C3 cycle. So take place in stroma of chloroplast in C3 plants. In the case of C3 plants, here I am taking plants. In the case of bacteria, within the chromatophores. In the case of eukaryotic plants, within the stroma. Go back to chloroplast. So it takes place within stroma. And That is in the case of mesophyll cells. C3 plants and C4 plants also. Simply remember the chloroplast in C3 plants and C4 plants also taking place within the stroma. That is common for both C3 and C4 plants. C3 plant means a plant with C3 cycle. C4 plant means with C4 and C3 cycle both are present. But the nomenclature of a C4 plant was based on oxaloacetic acid carbon number. C4 plants also having C3 cycle. C4 is additional mechanism. We will see at the time of C4 cycle. So C4 plants are with both C3 and C4. According to this statement, C4 plants also with the C3 cycle. Reason that is the only biosynthetic pathway during which CO2 is reduced into carbohydrate. Right? Now, what about a C3 cycle carbon reduction pathway design means prepared by, traced out by Melvin, Kelvin, Basham, and Benson. Now, what about the flow chart of a C3 cycle? So here to produce one glucose molecule, number of cycles of C3 required. Number of cycles, 6 cycles required. So here I'm taking all the reactions. They account for one glucose within one cycle and follow according to textbook. How many number of cycles given? We'll discuss that one at the end after completion of a total C3 mechanism. Okay, so here in C3 cycle, we divide all the reactions into 3 phases. First phase is a carboxylation. Accepting CO2 by concerned acceptor. We'll see what is that acceptor. And second phase is reduction phase. Reduction phase. And third one is regeneration phase. Carboxylation, reduction, and regeneration. These are the 3 phases. With the help of these 3, we can understand the cyclic flow of carbon reduction and the intermediates can be understood. So what about those reactions? Okay. So first phase is a carboxylation, right? So now here take 6 molecules of copy this one, 6 molecules of RUBP. RUBP. What is RUBP? Ribulose. R stands for ribulose. Okay, ribulose. 1 comma bisphosphate or diphosphate. Okay, so ribulose, ribulose. Okay, ribulose. 1 comma 5 bisphosphate. So actually it is a ribulose 1 comma 5 bisphosphate or biphosphate. 2 phosphates are present. First carbon with 1 phosphate. Fifth carbon with 1 more phosphate. So total number of phosphates 2. Biphosphate or diphosphate. So ribulose is a 5 carbon sugar, pentose sugar. It is a keto sugar. Keto group is present. C double bond O. Keto sugar. Keto sugar. So in C3 cycle, the acceptor of CO2 is a RUBP molecule. What about the number of carbon atoms? 5 carbon atoms. So 6 molecules of RUBP, ribulose 1 comma 5 bisphosphate, accepting. So 6 molecules of RUBP reacting with or accepting how many CO2 molecules? Here I'm taking 6 CO2 molecules. At a time I'm taking 6 CO2 molecules. 5 carbons, 5 carbon, 6 into 5, 30 plus 6, 36 overall. 6 molecules, 6 molecules. And here enzyme required is a Rubisco. Go back to biomolecules. Rubisco. Rubisco is a ribulose 1 comma 5 bisphosphate CO carboxylase oxygenase. C stands for carboxylase, O stands for oxygenase. So this enzyme is able to regulate two types of reactions: carboxylation, oxidation. If CO2 concentration is more than O2 concentration, carboxylation. Otherwise, oxidation or oxygenation. We will see that one at the time of photo respiration. So 6 molecules of CO2 accepted by 6 molecules of RUBP with the help of Rubisco enzyme. Most abundant protein in this world. Okay, so in the presence of RUBP plus 6 molecules of H2O, so RUBP is converted into, by accepting 6 molecules of CO2, it is converted into 12 molecules of this is 12 molecules of 3-phosphoglyceric acid, third carbon with 1 phosphate. So this is a diphosphate and this one is a monophosphate. It is a 3 carbon compound. 12 3 36. 30 plus 6 carbon 36. So 12 molecules of 3-phosphoglyceric acid. Enzyme is a Rubisco. So now in the presence of phosphoglycerokinase, enzyme name. So 12 molecules of 1 comma 3 bisphosphoglyceric acid is converted into 12 molecules of 1 comma 3 bi or bis or diphosphoglyceric acid. So this is a 12 molecules of 1 comma 3 bisphosphoglyceric acid. So here in this reaction 12 molecules of ATP consumed. So number of ATP molecules during this reaction, 12 molecules of ATP from the non-cyclic electron transport system. Here I'm taking like that. So 12 molecules of ATP hydrolyzed into 12 molecules of ADP. Okay, and remaining inorganic phosphates are added to that one. So 12 ADP plus 12 inorganic phosphates added to 12 molecules, each with a 1 inorganic phosphate. So monophosphate is changed into biphosphate or diphosphate. So here this reaction is carboxylation. Carboxylation with only one reaction. And this reaction is a first reaction of reduction phase. Carboxylation with only one reaction. That is ended with by the formation of 12 molecules of 3-phosphoglyceric acid. This reaction is a phosphorylation. This one carboxylation, phosphorylation. Monophosphate is changed into diphosphate. Now, next reaction of reduction phase. So 12 molecules of 1 comma 3 bisphosphoglyceric acid is reduced into reduction. This one is phosphorylation, reductive phosphorylation. Here phosphorylation, here reduction. During reduction, enzyme required is a glyceraldehyde. G is nothing but glyceraldehyde. Glyceraldehyde. glyceraldehyde 3-phosphate. What is a G3P? Glyceraldehyde. G stands for glyceraldehyde. 3-phosphate. Third carbon with the phosphate. Glyceraldehyde 3-phosphate dehydrogenase. In the presence of dehydrogenase, simply. Okay, so it will be reduced into a product. And reducing power. Here, what about the reducing power? The donor of reducing power is 12 molecules of Na DPH plus from non-cyclic electron transport system, oxidized into 12 molecules of NADPH, NADP plus, sorry, NADP plus. So reducing power. Okay, what about the total number of electrons? 12 into 224 electrons accepted by 12 molecules. So this is a reduction reaction. And now, whatever the number of inorganic phosphates added here are removed. So 12 molecules of inorganic phosphates are removed. So this is a little bit very complex and time-taking one. Without practicing all these intermediates, it is very difficult to understand. So several times try to practice it. Then only you can prepare the chart. So before learning so-called content part given in the NCERT textbook, please practice the flow chart. After commanding flow chart, you can understand what is going on within the C3 cycle. So here 1,3 bisphosphoglyceric acid is reduced dephosphorylation. So this is a reductive phosphorylation, reductive dephosphorylation. During non-cyclic electron transport system, out of 12 H2O molecules photolized, what about the number of ATP molecules? 24 ATP molecules. Out of 24, so for how many consumed? 12 consumed. And what about the number of NADPH plus produced there? 12. 12 consumed. Okay, right? Now, what about the product? So here 12 molecules of, 12 molecules. Here I'm dividing 2 molecules of G3P. Next, 2 molecules of G3P, glyceraldehyde 3-phosphate, 2 molecules of G3P, 2 molecules of G3P, 2 molecules of G3P. 1, 2, 3, 4, 5. Take one more. 2 molecules of G3P. For our understanding, how many are there? 1, 2, 3, 4, 5, 6. 12 molecules of G3P. So this is a pool of or group of glyceraldehyde 3-phosphate group. How many? 6 into 6 to 12 molecules of G3P. Glyceraldehyde 3-phosphates. Glyceraldehyde 3-phosphates. Total number 12 molecules. So out of 12 molecules, take this one. Out of 12, 2 are exported out. Exported into cytoplasm. Exported 2 are into cytoplasm, where these 2 trioses are converted into 1 glucose molecule. Reduction phase completed. So glucose is produced during reduction phase. What about the total number of G3P available? How many? 12 G3P molecules. Out of 12, how many are exported out? 2. So 2 by 12 exported out. So 1 by 6 if it is simplified. 1 by 6 exported out or not? So out of 12, 2 exported out. Calculate the percentage also. 2 divided by 12 into 100. That much percentage of trioses exported out. So 2 by 12, 1 by 6. 1 by 6, fourth of 1/6 part of, sorry, 1/6 part of G3P transported into cytoplasm to synthesize glucose molecule. So glucose synthesis is part of reduction. Reduction phase completed by the formation of glucose. Okay, so out of 12 molecules, 2 account for 2 out of 12 account for 1 glucose molecule. Place of formation of carbohydrate is glucose is within the cytoplasm. So now how many are available? Only 10 are available. Remaining 10 are involved in regeneration phase. So now how many are available? 10. 10 divided by 12. So 10 by 12 for regeneration. Regeneration of 6 molecules of RUBP. That is a 5 by 6 available for regeneration of RUBP. So 10 divided by 12 into 100. That much percentage of G3P molecules are involved in regeneration of 6 molecules of REBP to run the reaction again and again. Out of 6 CO2 uptake, what about the number of glucose molecules? 1 glucose molecule, right? Remember this probability. How many G3P available total? 12. Out of 12, how many exported out? 2. So 12 G3P molecules. Out of 12 G3P molecule, 2 molecules of G3P for 1 glucose molecule, and remaining 10 G3P molecules for regeneration of 6 molecules of RUBP. So how many are involved in regeneration phase? 10 molecules. Okay, so G3P is a glyceraldehyde 3-phosphate first sugar produced during C3 cycle. G3P is a sugar. Glyceraldehyde, aldehyde. Okay, remember. Now regeneration phase. So now during regeneration phase, second one, fourth one, for understanding only. 2 plus 2, 4. Isomerized into 2 molecules of DHAP, dihydroxy acetone phosphate. 2-DHAP, dihydroxy acetone phosphate. Here enzyme required is a triose, triose phosphate isomerase. Triose phosphate isomerase. So in the presence of triose phosphate isomerase, 2 plus 2, 4 molecules are isomerized into its isomer, that is a dihydroxy acetone phosphate. Remember this one. So that is isomerization is the first reaction of regeneration. First reaction of regeneration is isomerization. Next, now 2 molecules of G3P, 2 molecules of DHAP. Condensation, getting together. So enzyme required is the aldolase. Aldolase in the presence of aldolase enzyme. Okay, right? In the presence of aldolase enzyme, condensation reaction. 2 molecules of DHAP. This is a 3-carbon compound. 3-carbon compound. That is also 3-carbon compound. So what about the product of condensation? 2 molecules of fructose 1,6 bisphosphate. It is a 6-carbon sugar. Sorry, 6-carbon sugar. So the product of first aldolase is 2 molecules of a 6-carbon bisphosphate, fructose 1,6 bisphosphate. So now splitting of dephosphorylation, elimination of inorganic phosphate. Elimination of inorganic phosphate. 2 inorganic phosphates removed. Enzyme required is a fructose 1,6 bis phosphatase. Fructose 1,6 bis phosphatase. Phosphatase. So nowadays, convert into two molecules of fructose 6 phosphate. It is monophosphate. From each 6 carbon sugar, one phosphate is removed from first carbon atom. Nowadays, monophosphate. So two molecules of bisphosphate is converted into two molecules of monophosphate. Here enzyme is a bisphosphatase, right? So that is about fructose 1 comma 6 bisphosphate converted into fructose 6 phosphate phosphatase enzyme. Now two molecules of fructose 6 phosphate. Now condensing with start from here, condensing with two molecules of G3P in the presence of TK, trans-ketolase. Here I'm writing TK, there is no space. TK, trans-ketolase. TK is a trans-ketolase. In the presence of trans-ketolase enzyme, two molecules of fructose 6 phosphate condense with the two molecules of G3P. So here, what are the products? So products are two types of products, two kinds. What are they? Two molecules of xylulose 5 phosphate. Xylulose, XYLULOSE, xylulose 5 phosphate, and two molecules of erythrose 4 phosphate. 5 carbon, 4 carbon. Xylulose 5 carbon. Erythrose 4 carbon. Monophosphate, monophosphate. At fifth carbon atom, 1 phosphate. Fourth carbon atom, 1 phosphate. So erythrose is a 4 carbon sugar. This one is a 5 carbon sugar. So that's about a condensation reaction here. See, aldolase, phosphatase, transketolase. Now, two molecules of erythrose 4 phosphate. Condensation between these two. Okay, so enzyme required is a same aldolase. Second time involved. Aldolase. First involved, second involved. Right? So now two molecules of erythrose 4 phosphate 4 carbon reacting with the two molecules of DHAP, that is 3 carbon. So what about the product? Here product is two molecules of S1 comma 7 bisphosphate. It is a 7 carbon diphosphate. S is nothing but a pseudohaptalose, pseudo or pseudohaptalose. So two molecules of pseudohaptalose diphosphate is produced, like this one, diphosphate, diphosphate. This one is a 6 carbon, this one is 7 carbon. So what is the product? The product of second aldolase, second time involved aldolase is the two molecules of pseudohaptalose, haptose, 7 carbon diphosphate. So now dephosphorylation, elimination of two inorganic phosphates. Okay, here enzyme required is a phosphatase. What is that phosphatase? Completely pseudohaptalose 1 comma 7 bisphosphatase, like this phosphatase. So it is a dephosphorylated into two molecules of pseudohaptalose 7 single phosphate, monophosphate or phosphate. Okay, so from first carbon atom, one inorganic phosphate is removed, two inorganic phosphate removed. So it is a pseudohaptalose 7 phosphate, monophosphate, like this, right? Phosphatase enzyme. And once again, aldolase, phosphatase. Go back, aldolase, phosphatase, transketolase. Now, what about our next reaction? Now, 2G3P reacting with two molecules of pseudohaptalose 7 phosphate. Here enzyme required is TK, transketolase. Enzyme is TK. Second time involved, TK, transketolase. Okay, so here what are the products? Two molecules of same like this. Cellulose 5-monophosphate. And next one, two molecules of ribose 5-phosphate. Okay, so transketolase, two types of products, xylose, erythrose, 5 carbon, 4 carbon. Next, here transketolase, two types of products, xylose, same, common, 5 carbon. Next, ribose, 5 carbon. See here, 5 carbon, 4 carbon. This one, 5 carbon, 5 carbon. These two same, these two different. All these are sugars only. Okay, right? So what about the total number of xylulose 5-phosphates out of all these reactions? Two molecules. Now, four molecules of xylulose 5-phosphate. 4 molecules of xylulose 5-phosphate are changed into 4 molecules of ribulose 5-phosphate. Here enzyme required is epimerase. So in the presence of epimerase enzyme, 4 molecules of xylulose 5-phosphate epimerized into 4 molecules of ribulose 5-phosphate monophosphate. Next, 2 molecules of ribose 5-phosphate isomerized into 2 molecules of ribulose 5-phosphate. 4 plus 2, how many? 6 molecules of ribulose monophosphates. Enzyme required is isomerase, that is actually ribose 5-phosphate isomerase. Okay, right? So what about the total number of ribulose 5-phosphates? 6 molecules, 2 plus 4, and it is monophosphate. What we need? Biphosphate. To run C3 cycle, ribulose monophosphate must be converted into biphosphate. So now phosphorylation in the presence of ribulose 5-phosphate kinase. 6 molecules of ribulose monophosphate is phosphorylated into 6 molecules of biphosphate. Here 6 molecules of ATP utilized, and that is converted into hydrolyzed into 6 molecules of ADP. And during this reaction, what about inorganic phosphates? How many inorganic phosphates? Here I'm writing 12 molecules of 6 molecules of 6 molecules of inorganic phosphates coming from hydrolysis of 6 ATP molecules. So 6 inorganic phosphates added to these. So monophosphate is converted into diphosphate. So RUBP is regenerated to run the reaction again and again. So carboxylation is a lengthiest process, time consuming one. A number of reactions are there, right? So that is about a C3 cycle.